There it is, yeah. So good morning, everyone. I know it's getting close to lunch, so I hope you're hungry to learn more about Elastrin Therapeutics. I've been thinking about this joke a long time. What you can see on our title slide is saying a lot about our company. We're restoring the elastic of life, and we're doing that by regenerating damaged elastin. What you can also see on the slide is the beautiful hometown of our facility. That's Greenville, the waterfalls, so everyone to South Carolina as your next holiday trip. So before we get going, just a little bit about elastin and what we're doing. And everyone in this room has damaged elastin, and at least 1 billion people in the world have damaged elastin, and that's linked to the fibers that degrade once we grow older. And what we have developed, and when I say we, it's definitely this gentleman here in the front, Professor Vaivahari, is a nanoparticle that has a targeted approach and binds only to damaged elastin. And I know, for example, I think it was Alex yesterday that said, well, targeting is maybe not that important. For this one, it is very important. So what is elastin? I don't think it's the most popular of tissues in the body, although it is one of the most important ones. It's a main component of our lung and our blood vessels, also of our skin, and it allows you to pump around blood and oxygen. The problem is that it's produced only once in a lifetime. It's a bit more complex than what's on the slide, but just to give you an overview. And the body doesn't really have the capacity to restore it once it gets damaged. So you can see it quite nicely on this picture. On the left side, you see healthy elastin. It looks like a rubber band. And on the right side, you see in skin, for example, it's very obvious. It snaps, and where it snaps, that's where the inflammation starts. And with that inflammation, a second process gets kicked off, and that's the hardening, the calcification. In itself, this is a chronic disease, and that leads to more chronic diseases, especially the cardiovascular system, but also in the lungs. And that's why we believe that targeting damaged elastin is a source for new therapies. And we can do a quick experiment, everyone, on what is elastin and damaged elastin at work. So everyone hold out your hand like this, pull it a bit to the back. Now try and locate someone who's a bit older than yourself and do it gently because you don't want to upset everyone. Then pull back your skin for about 3 seconds, let it go, and look how long it takes to go down. You can do it a couple of times, hold it up long enough. And what you will see with your elderly population in the room is that it goes a bit slower back to its original state than with the younger people in the room. This is damaged elastin at work. In skin, it's maybe not the biggest problem that we have wrinkles, but in the cardiovascular and the lung system, it's of course very important that it keeps functioning. So if we take a look at our product, and this is of course the most important thing why we're here today. We have a nanoparticle with a drug load, and we have several drugs that we load into this nanoparticle. You can see it as the red dots on the picture. First drug that we put inside is EDTA. EDTA binds calcium, the source of the calcification. So this is to really chip away that harmful calcification that builds up. Our second drug in development, and I'll tell you a bit more about the third one later, is PGG. It's a natural compound, our own compound developed, and it allows you to restore the damaged elastin. The orange part of the picture is a human albumin nanoparticle. As said, it's a safe vehicle. It's biodegradable. We all have it in abundance in our blood, so it just gets degraded, and you pee it out. And then on the surface of this nanoparticle, and this is really crucial, that's where the targeting comes from. And it's an antibody. We call it flexipsomab. After a while, it goes easier to say it. Beginning is quite complicated. But this is the one that allows you to target damaged tissue alone. And it's a combination of a vehicle, of a vessel, that can transport the drug, and the targeting on the antibody that allows you to have much higher concentrations of your drug only at the diseased tissue. And this is again another depiction of how you will see it at work. You start with healthy elastin at number 1 when we're young. When we grow older, it gets damaged for a number of reasons. We know radiation, pollution plays a very important role. That triggers the calcification. That leads to hardening of the arteries. And we get these arteries from amputated patients. We're on top of a cardiovascular clinic, and they are rock hard. They look a little bit like this. So they don't look like a red artery anymore, but they're you can tap on the table with it. And that, of course, does not allow you to pump around blood. What we are doing is we're stopping that inflammation, we're reversing that calcification, and then we amplify this effect by adding PGG to the mix. PGG allows you then to restore the elasticity of your arteries or your lungs. Again, this is curative. This will solve a disease. What I'm also 100% convinced of is that we can use this preventive. Maybe everyone above 30, 35 would come in once a year to restore its pipes, so to say. We have done a lot of animal studies, and with a lot, I really mean a lot. And I think it led us to one of the biggest challenges in the last years. We have great data on a lot of things, so what are we going to do now? And I think some of the other presenters had the same issue. It is important to focus, because else you won't get anywhere. That's my belief. So we focused on the cardiovascular calcification. There's many more publications. If you want to read them, we'll put them all on our website, so you can have a look. And one of these studies, I think this is actually the one, just to show you a little bit of scientific data, is on the left side, you can see administration of PGG and EDTA, and that leads to a 3-fold reduction in cardiovascular calcification. It also leads to restoration of the elasticity. And when keeping these mice alive, this is not on the picture, you see that they survive, and other ones would die. So again, talk to Naren about all the great stuff he's done in his lab. If we look at it on a bit deeper level, these are harvested arteries of those animals. You can see on the left side, they show up severely calcified, and on the right side, the calcification is almost completely invisible. And you can see it with other stainings as well. There's typical ways to show, for example, not only elastin damage, but also decalcification. Something we've recently did, and again, I say we, but it's actually Sonia here on the front. I like to take credit for what these great people are doing, is we've in-licensed a messenger RNA. And a messenger RNA, that is not a vaccine, but it's a therapeutic one. So it will allow you to produce more precursor of healthy elastin. It provides fuel for the regeneration. And you can see it quite nicely on this picture as well. You start on the left top with the messenger RNA. It goes into the cell. You start producing more of these precursor, and they get then they come together and form healthy elastin or new elastin. And the advantage of such a synthetic messenger RNA is the same one that, for example, the BioNTechs and the Modernas use, is that you can fast and effectively increase your production of your precursor. On top of that, it doesn't go into the genome, so there's safety issues or less safety issues. It only works for a while, so you don't need to think about long-term safety issues. There's very good IP protection on it, and we're adding some more. And the results that Sonia got with this are quite impressive. Both in cell lines, you can see de novo synthesis, new elastin being formed. You saw that in skin tissue, and you saw that actually in also live animals. So we're building on that. It's a bit earlier than our other projects, but it's catching up. If we put all that in a mixer and shake it a bit, this is what comes out. We have 3 pillars in our clinical development. And the first pillar, of course, that's our EDTA compounds. It's the earliest, so that's why we started with it. It's the furthest along. And the second compound is PGG, or the second pillar is PGG, and the last one is the messenger RNA. We're thinking of combining it with our second pillar. Maybe there's some things we can do with it alone. It is, like I said, it needs some maturation. And what is crucial to our clinical strategy is that we pair up an orphan indication with a broad indication. And we call them Trojan horses, just like, when the Greeks were attacking Troy, we tried to get into the development much faster with first going for an orphan indication, not only saving a lot of money, I think that's important, but on the other side also moving much faster to our broad indication. And we're going to do that about 5 times, and that also then leads to this great priority review vouchers that you, for example, have rare pediatric, orphan drug status. It gives you a lot of tools as a company to go much faster to patients than without it. And on top of that, we're also working on a data master file. It's again a regulatory tool to get faster into the clinic, to look at to show the regulator that your platform works and that it's safe to use. From a timing point of view, as mentioned, our first compound is well ahead. We're starting our clinical trials next year. Second one is about 6 months delay, and the third one again 6 months. So that needs some work. We know for our first compound, we've talked to the regulators, the orphan status is both confirmed in with the FDA and the MHRA. We're going for a dual approach there as well, first in the UK and then in the US. But it doesn't stop there, and I think that's important. Again, I like to iterate that it's a platform technology without losing focus. We have great results in chronic lung disease to regenerate lung tissue. There's a bunch of other indications, for example, in the eye, macular degeneration is one where we see that we have potential. None of these, I believe, we will do ourselves, but it's great opportunity to team up. And again, I think it was Mike who said a lot about the business case around cardiovascular disease, so I don't need to say a lot about it. It's quite obvious cardiovascular disease is the #1 killer in the world. It's rising in a lot of countries. It's going down in Europe, but it's going up anywhere in the rest in the world. It's a huge market. We're looking at capturing a part of that will be financially very attractive. But on the other side, and this is also very important, for all the indications that we had on that slide, there's almost no treatment option available. And the treatments that we have are symptomatic. So again, they're not about solving the disease, they're just about solving the symptoms or curing the symptoms. And there was a long time that big pharma stepped away from cardiovascular disease for obvious reasons. I mean, the clinical trial cost is, of course, one, but we see that it's slowly coming back. There was a big deal end of last year with CSL and Vifor Pharma and Sanofi. They all got together. We had AbbVie a while ago. Daiichi Sankyo is now there as well. So it shows that at least also the larger companies are looking towards cardiovascular disease. From an IP portfolio, and this is of course very important as a company, is that you're safe and that you know that you will be able actually to bring something to market without anyone else stealing it. We have 3 patent families. The first one on our, not surprisingly, on the 3 parts of our therapy. The first one on the drug itself. The second one on using these nanoparticles to target damaged elastin. And the last one, and you see they go a bit consecutive because that's also how the development went. It's on the antibody itself. It's being applied all over the world. And because it's such a key asset of our therapy, we do believe that this is one of the most important IP parts that we have. From a timing point of view, well, 21 is all over yet. 2022 is also almost all over yet as well. But I just wanted to show that we actually only got started beginning of last year with going into an extra drive. The company was founded a bit earlier, and we had a lot of great grant money supplying the R&D, but it was with the first funding round that we really got into a acceleration. We built our US team, we manufactured our antibody, we're toxicology testing, it's all happening. I think it's a completely horrible time in the development because everything that can go wrong goes wrong, but we keep on pushing to start our first clinical trial mid of next year, and that will then be consecutively followed by other ones. For me, very importantly, it says a phase 1 trial, but if it were up to me, I would never do a phase 1 trial. I think it's ridiculous to dose healthy volunteers, apart from that they earn some money with it. We're really going to try and do a phase 1/2 combined study. Having healthy patients, for example, in that study, you can immediately see safety, but also efficacy. We're raising funds next year, so luckily we aren't too rushed now in this crazy year with the economical recession. And we also believe that 2025 will be pivotal for us. We'll have phase 2A data, we'll know if it's safe and it works, or kind of works. And I think at that point, I would love to do an IPO. I mean, that's one of my personal dreams, to ring the bell. Yesterday we saw Vlad saying that was quite the experience. So I think this is also something with clinical evidence that you can really go to the market and say, look at our company, what about spending some money yourself? So we have a lot of people from Elastrin here for this conference, about 14. I think that's very nice because they can talk to everyone. We have, you know myself in the meantime. You've been looking at my face for a while. Naren, here up front, he's the scientific founder of the company, together with Charlie, Professor Rice, who developed the antibody, and Douglas. We have Sonia, who just joined the team. She comes from the middle of Germany, and she is the one who developed the messenger RNA, so we thought, well, if we get the messenger RNA, we'll better also get the person who worked on that. We have Alyssa, who works in our lab in South Carolina, and Patrick, you're on the slide. I hope that's okay for you, because we do feel that you're part of the team. We also founded a new scientific advisory board. I think it's extremely important from a clinical point of view that, you know, I believe that it works, and maybe some other people, but you need to have a broad agreement in the scientific community. Some of them are here as well. We have Professor Frank Rutsch, Dr. Klaus-Dieter Langner is here, Professor O'Neill, so do please talk to these people if you have any questions, and I hope they can condone everything that I said. This leads me to my last slide, just summarizing what I said. It's a lot of information, but I think what is crucial and the one learning that you should know is that, and that sets us apart from many other companies, is that we're reversing the disease. We're not trying to solve symptoms. Of course, that's the first step that you want to have, but we're trying to go back to a stage when you were still healthy. And we do that by restoring damaged elastin, working on the calcification first, then restoring the damaged elastin. I think we have a great team, and I'm very happy that, like I said, that we're all here today. I think one of the most important things in a company as well is that you have a regulatory strategy, that you'll know that you'll get to market, to the clinic as quickly as possible. So we're following this dual approach with the United Kingdom and the US. And we're here, for the moment, not raising funds as mentioned, but always looking for potential future investors. I think that's important. Also other collaboration partners. If you have an idea, even if it's really crazy, just come to us and we'll see if we can make it work. Typically, this is where I end my presentation, but just for you guys, I have a premiere, something crazy new that nobody knows yet, and it's in the form of a little video. So if we could get some sound. Yeah. That was it. So thank you, Charlene, for actually building this video again. I don't want to take your credit.
Oh, thank you for this fantastic presentation.
Thank you.
This has to work. We all need it, so make sure it's happening. Okay, do you have a question for Matthias in the audience? Please raise your hand. Yeah, you guys are starving. Yeah, that's one.
Hello, thank you very much for the nice presentation. So I'm excited, and don't find my question as criticism or negative, but my question is, there have been 5 or more clinical trials that used EDTA for decalcification, so chelation therapy, and all 5 failed. So why do you think your targeted delivery would be more efficient?
Yeah, I think the answer to that is a bit double. I do believe that there are some calcification studies that actually work. Sanifit, for example, they show actually very good results. Also, the trials that Professor Lamas is doing, NIH-funded trials, he uses EDTA in high concentrations every week, so for a year, and you see that the decalcification, or at least the limbs, are being saved. Decalcification is not an easy topic, right? It's a complex process. We know it's not as easy as just, you know, well, if we stop it, then it's over. we can only trust, I believe, our animal data, and we see that we've tried it a number of times, also in human tissue, as much as possible, that the decalcification works and that you see almost complete removal of that. And that then allows you to restore the blood flow and also to get the elastin regenerated.
Thank you.
Okay, I think we are good. Thank you, Matthias.